Singularity.
My name is Nicola, aka Socrates, and you're watching Singularity One-on-One. Singularity One-on-One is a regular podcast feature Singularity weblog, where you can go and listen to it or download it in full. As always, you can guys help me make this show better in one of two ways. You can simply go to iTunes and write a brief review, or you can make a donation on my donations page. Today, my guest on the show is Ken Hayworth. Ken is the person behind a very unique and new cutting-edge way of brain preservation. And that's, of course, the reason why I wanted to bring him on the show. So without further ado, let me welcome Ken and say hi, Ken, and welcome to the show.
Hello. Thank you very much for having me on.
Fantastic. Ken, I kind of messed up your introduction here a little bit, so let me ask you to please introduce yourself. What do you do, and so on?
Yeah. So let me correct one thing. So I don't actually do brain preservation myself. I actually my background is in electron-microscopy of neural tissue. Mm-hmm. So I invent different techniques for mapping brain circuitry at the nanometer scale, and in that process, I come across these standard techniques that people have used for preserving brain tissue, very small pieces of brain tissue, and I have realized, along with other people, that if this could be expanded, these standard techniques, if they could be expanded to a whole human brain, then that would be a quite revolutionary thing to have the ability to do, to preserve an entire human brain for future technology to use and to possibly bring back the individual. And so I started the Brain Preservation Foundation a few years ago. We have a Brain Preservation Prize, and basically it's a challenge to the scientific community and also the cryonics community to develop a technique, any technique that can preserve an entire human brain at the level that neuroscience says is required to preserve all the memories and personalities of an individual, namely the synaptic connectivity of the brain. And so I get a chance to work with great groups of people that actually do research in brain preservation.
Mm-hmm. So I should probably have called you a champion of a new and different way of proposal for brain preservation. Now, let me ask you this. Do you have an official, what's your full-time day job title? And where do you work, and do you have any scientific background?
Yes. So my full-time job is I'm a senior scientist at the Howard Hughes Medical Institute at Jamelia Farm Research Campus. Before that, I was at Harvard University doing a postdoc, again on electron microscopy. And I have a PhD in neuroscience from the University of Southern California in cognitive neuroscience, studying the visual system of human beings and modeling it, the computations that are done within it.
That's fantastic. So before we move on to the meat of the matter here, as it were, I'd like to roll back the tape a little bit and ask you, Ken, what was so interesting about, electron microscopy, and why did you get inspired or decide to work in that field?
Yeah. Well, if you wanted to go way back, I think when I was a kid, I was absolutely I absolutely wanted to go into space. I think there's a lot of people out there that are like that. I love Star Trek episodes, and I absolutely wanted to get off of this rock and go explore the moons of Jupiter or the next star system or something like that. And as a little kid, I kind of made a pledge to myself that I would eventually do that. I haven't gotten anywhere close to that, but when I got old enough to look into the actual science behind going into space, and especially going to the nearest star system, I was really depressed that it took so much technology. And maybe I was about 15 at this time, and I came across, I was reading books like the Starflight Handbook and things like that would talk about antimatter rockets, and I came across this relatively new field of neural networks. Mm-hmm. And it dawned upon me, like it dawns upon a lot of people, I suppose, that the problem with spaceflight is human beings. They're too heavy. They take too much. They take too much resources, and that if we are just information, which is what neuroscience and cognitive science tells us, then we have sitting in front of us a light speed travel method, because our radio waves already go at the speed of light. And so I pretty much switched gears. I said, I found it. The way of getting to the stars is by neuroscience. It's not by developing a multi-generation humongous spacecraft that costs trillions of dollars, with engines that we don't even have the physics to begin to understand how they could work. It's right in front of us. It's neuroscience. And if we can understand how the brain works and how to extract what we really are from the brain, then encoded in ones and zeros, we'll be able to transport that via radio waves to, in a very efficient way, to any receiver that we put out there on any of these systems.
Mm-hmm. So is it fair to say that ultimately your goal is to sort of travel the galaxy or the universe?
I think it's fair to say that, but I think it's traditional that people get interested in one thing, it leads to another thing, and then they realize that they're even more interested in that the thing that it led to.
Yes.
And so it is very exciting still, the idea of space travel, and I would love to experience that. But the possibilities of actually understanding how a human mind works, putting it into a different substrate that doesn't have disease, doesn't have limitations, that is an even greater frontier than space travel. Mm-hmm. And I hope to get to see both of those.
Yeah, I have to agree with you entirely that I think that the most likely method of colonizing, the galaxy and perhaps the universe would be not within our bodies, which can only take so many Gs and, require so much energy and space and so on to sustain them, but with mind uploading and with perhaps beaming ourselves across the universe. But you gave us a little bit of a glimpse on your other motivation, on your additional motivation to do that work, which it seems to me that now is kind of taking over. So why don't you tell us a little bit more about that you just mentioned, the conquering disease and so on?
Yeah, well, so I'm one of many people that believe that the future is going to be, we're going to get better and better and better in technology, that we are going to cure diseases, that we are going to have fantastic levels of technology that can scan an entire human brain, can repair things, biological damage that we wouldn't dream of being able to repair today. And so, but at the same time, I look around and I see how fast we're moving toward that, and we're not moving fast enough for people today. Mm-hmm. I am not one of the people who believes that aging will be cured in my natural lifetime. I'm not sure if it will be cured in my kids' natural lifetime. I am skeptical.
So let me stop you right there for a second. So you're skeptical on curing aging in your lifetime and perhaps even your kid's lifetime, but are you equally skeptical or not so skeptical about being able to mind upload people in your lifetime?
Yes, I am.
Yes, meaning you're skeptical or optimistic?
I'm sorry, one more time.
Yes, you're optimistic or you're skeptical?
I'm skeptical. I think it's uploading a human being is an incredibly difficult task. If we wanted to get there, it would still take decades and decades. We're not exactly putting all of our resources into a grand Apollo project to upload a human being.
Yes.
So no, we're probably many decades away from uploading a human being. We're probably many decades away from curing aging. There are so many diseases that would have to be conquered. And so I think I'd like to be proved wrong, obviously, but I think that brain preservation, like cryonics, the idea of cryonics is simply that we accept that we will have advanced medical technology in the future that will be able to cure whatever has happened to us today, but we can't get there unless we go into some kind of state of suspended animation. And so the question is really, where do we put our resources? And from what I've seen, the problem of curing aging is enormous. The problem of mind uploading is enormous. The problem of putting somebody in suspended animation is not enormous, especially if you come to grips with the idea that we should think outside the cryonics box. We don't have to think about technologies that are just a hair's breadth beyond what we have today and think, okay, well, this is almost good enough to just be rewarmed, and the person walks off the table. If we can look at technologies that are preserving, using chemical preservation, the structures of the brain, then we have a quite advanced, since the 1950s, we have been doing very good chemical fixation and plastic embedding of brain tissue at the so that we can look at the brain at the electron microscope level. I mean, by definition, this has to be extremely good preservation because all of the textbooks since the 1950s have been saying this is how the brain works. This is what the synapse looks like using these chemical preservation techniques. And so if we look at that and say, wow, all that we have to do is take that existing technology and apply it to larger brain volumes, a whole human brain, then that's the suspended animation that we might have been looking for. Mm-hmm. Because that will allow us to put somebody on the shelf in a non-decaying state that can last literally probably for millions of years in that type of chemical preservation, plastic embedding environment, to wait the technology that can bring them back. Mm-hmm. And this is especially important because we can now see the types of technologies that could possibly be bringing somebody back from that chemically preserved and plastic embedded state.
Ken, I want to come back to the topic here and talk more about your proposal for chemical brain preservation and the difference between that and cryonics. But before that, I want to grab one point that you made before that and talk a little bit more about that, and that's about the fact that you're skeptical about mind uploading. So I had Dr. Randall Kuna on my show, and according to him, mind uploading is not science fiction anymore. And while he also agrees absolutely that it's a humongous undertaking, he doesn't dismiss it outright that it might be possible. In fact, he says that it probably would be possible within our lifetime, if I remember correctly. Also, we have at least two other. So Randall Kuna works on what he calls whole brain emulation. Now we have people like Dr. Henry Makram, who works on whole brain simulation project and who wants to raise $1 billion and who believes that it is entirely doable. I think in our lifetime. And finally, a third project that comes to mind is the IBM Synapse project that's funded by DARPA and I think is led by Dhametra Motha. And I think IBM actually has a much more ambitious timeline, and they're aiming to map an entire brain by, I think, something like 2018, which is very ambitious. So, therefore, doesn't it seem that, you know, a number of people are pursuing those different ways of accomplishing that task, but within a decade or two, clearly within our lifetime? Doesn't that give you any sign or evidence to hope that you might be wrong?
Well, I can always hope that I'm wrong. The other thing is, Randall's probably, he's a healthier guy than I am. Maybe he works out better and stuff. So it could be a difference in lifespan, or assumed lifespan. In any case, if you look at these technologies, so the DARPA Synapse Project and Henry Markham's project, to my understanding, those are computer simulations only. They don't address the biology side, and so we really have what Randall is talking about. Or what he may be talking, he talks about a lot of the different technologies, but I think the one that you're referring to is the idea of having a really massive functional recording of all the neurons in the brain during somebody's awake behaving experience. And so that could give you the information to create a simulation. So if we look at these different technologies, let's say, I'm going to pick three that I think are relevant to this type of mind uploading. So you could get to mind uploading by doing whole brain recording of every neuron in a brain, and then doing the simulation on a computer. That technology is advancing, but I would not say that it is going to be in a few decades. It is incredibly difficult. I mean, the place where they are now, the things that you would have to be able to do is at the very least, you'd have to get advanced gene therapy to work, because you're going to have to use advanced gene therapy to get the recording instruments into each of the cells. And so there are a lot of different technology leaps that have to occur to get that to work. And the hardest is definitely not the computer simulation of Markham, etc. There is, well, let's just take the other example. Let's just say two. The other way of mind uploading is to get the connectome of an individual, getting the electron-microscopy of the entire circuitry of the brain, mapping that out and understanding that in such a way that you can get it into a brain emulation. Again, this requires a lot of new neuroscience information. This requires all the mapping technology exists, and the neuroscience that will come from that mapping technology. And again, the brain emulation. So I can't bring myself to accept that that's going to be done in less than a few decades. Mm-hmm. I'm too skeptical of that. It seems too difficult, all of those problems. Plus, if you look at the first part of that procedure, the first part of that brain emulation, that brain mind uploading procedure, is exactly some type of brain preservation. Because you have to, at some point, put the brain into a state that can allow it to have high-resolution imaging performed on it. Mm-hmm. And the only way that we know how to do that is chemical fixation and plastic embedding today. Right. So it really comes down to, you can say, is it going to be 2 decades? Is it going to be 5 decades? Is it going to be 20 decades? It doesn't really matter. It comes down to, it's going to be, really, really difficult. And most likely you're not going to get there. The best way to put your resources, the best place to put the resources, in my mind, certainly, are to go into the preservation steps so that you have time to get to that technology. Mm-hmm. So I'm not doubting that the technology is going to get here. I'm just doubting that it's going to get here at any of the timescales that its most advanced advocates are saying. Mm-hmm.
Yeah. Well, I think you're making a pretty good argument. At the same time, though, history, some people say an expert is someone who can tell you what is not possible.
Yeah.
And history is full of examples like that. What comes to my mind right now, for example, is the president of the Royal Society, Lord Kelvin, I think, who in 1896 famously argued that heavier than air aircraft are absolutely impossible to create. And then 6 years later, 2 bicycle makers from the United States, the Wright brothers, who were bicycle makers, they were not really scientists, proved him wrong.
Yeah, absolutely. So, and there's plenty of those examples, and I would love to be, I'd love to be proved wrong on this. Yeah. But the thing is that most of our citizens out there, don't even go with the first argument. Mm-hmm. They would say this will never happen. That mind uploading is impossible in principle. And I think that that's particularly, that is on the order of the type of skepticism that we need to argue against.
Yeah, I want to grab that point right on here and actually read a passage of one of your papers that I read as preparation for this interview. It's from actually page 3 of a paper called "Killed by Bad Philosophy: Why Brain Preservation Followed by Mind Uploading is a Cure for Death." And it was written by you in January 2010. So the paragraph goes like this. Our grandparents had the technology to preserve the precise neural circuitry of their brains for long-term storage. The best science of our grandparents' era stated unequivocally that this unique patterning of neutral circuitry was the seat of the self. In it was written all memory, skills, and personality. Our grandparents seemed to grasp the quickening pace of technology and understood that full brain scanning and simulation was around the corner. Why then did Grandpa and the rest of his generation reject brain preservation in mind uploading as a means of overcoming death? And after considering the evidence, our grandchildren will come to the sad conclusion that we were killed by our bad philosophy. No matter how clear the science was, we simply could not really accept the fact that we were physical machines. Yeah, so I think that's a very strong and very good paragraph, so I'd like you to elaborate a little bit more and unpack it for us.
Yeah, this is from an essay that was, it's a part of an essay where it's looking back from the year 2100, so 100 years from now, 2110, I guess, and basically looking back with hindsight and saying, how should resources have been allocated, and how should people have viewed their options for getting to the future? Because essentially, we all should be jealous of people in the future. 100 years from now, a couple hundred years from now, people are going to be, just like people 200 years ago should be jealous of us, flying airplanes and living healthier lives and have antibiotics and wonderful things. And people 200 years from now are going to look back and say, death was horrible, and I'm surprised people could stand it. They should have been concentrating all of their time and resources on overcoming this great human limitation of disease and death. And if you look at it in that perspective, you can say, okay, so what were the resource allocations that people should have made today? And I think that from the future's perspective, they will see, brain preservation is the logical lifeboat that people have access to today. It is a technology that people can develop relatively easily. It doesn't require new physics. It doesn't require new biology. It requires a little bit of surgical technique advancement and some protocol development, and that's all. And the fact that nobody was doing that research. So I should correct. I'm not proposing chemical brain preservation as a cryonics alternative. I am proposing that, but I'm certainly not the first person to propose that. Of course, its roots go way back in the cryonics community as well. And there's a guy named Charles Olson that wrote a paper probably 20 years ago that made it very explicit that this is something that should be developed. So it is certainly the case that other people, in fact, Carl Sagan has a wonderful piece in his book Broca's Brain that is very tantalizing about the possibility of chemical brain preservation. It's surprising that he didn't follow up on it more.
Mm-hmm.
So go ahead.
So let me ask you then, can you walk us through the sort of the details or the procedure of chemical brain preservation as you see it?
Yeah, absolutely. So chemical brain preservation has been done as part of electron microscopy for since the 1950s. What happens is that the animal that you're going to look at under the electron microscope eventually, the animal is alive. You put them under anesthesia, and then you open their chest cavity up and go directly into their heart, their left ventricle, with a needle that will perfuse a formaldehyde and glutaraldehyde. It's similar to embalming fluid, but it's more aggressive than embalming fluid. That glutaraldehyde is a chemical that actually has two reactive groups that binds proteins together. It basically glues the machinery of the cell together to almost instantly prevent decay. It obviously kills the organism. It is one of the most toxic chemicals around, but it kills it in such a way that it is basically just sticking proteins together. That means that you don't have a lot of structural changes in the cells, and the proteins that should be at some place within a neuron usually stay within that place in the neuron. And it happens so rapidly. This is one of the advantages of this versus cryonics, by the way. Just a little off the track. The amount of time that you go from a decaying to a non-decaying state, a static state that you can that is stable, is just probably less than a minute in chemical brain preservation. Whereas in cryonics, you have to go through hours of slow perfusion so that you can eventually get enough prior protective agent in to get to a low enough temperature that you stop by temperature the decay processes. Mm-hmm. So if you're the idea of using chemical means to really hammer the decay processes into a stable state is a real key advantage that I think gets lost in this discussion at certain points. In any case, once you perfuse through the bloodstream with this glutaraldehyde solution, you now have something that is stable for days and days and days at least. You're not going to be able to leave it in that type of a situation because there's still very slow chemical reactions that are going on. So what people will do in the traditional method is that they will then take the small region of the brain tissue that they're interested in, they'll dissect that out. This is traditional electron microscopy. They will put that in a chemical called Osmium Tetroxide, which stabilizes the lipids of the membranes. And at that point, all the major molecular species, the proteins and the lipids, are in a stable state, and that allows the final step of extracting the water and replacing it with plastic to occur. Mm-hmm. If you didn't stabilize everything before that step, there would be a problem. So once you extract the water, replace it with plastic, you can cure that plastic. What you get is a solid block of no water tissue that can literally it is perfect fossil. It is like an insect caught in amber, except at all levels, the amber has preserved everything. So it's just a perfect fossil, and it could last for literally millions of years.
So basically, you're turning us into human fossils.
Essentially, yes. Essentially, yes. So, so the only part of that procedure that doesn't work on larger brains are the one that has mainly been said in the scientific literature not to go to larger brain tissue, is the osmium tetroxide step, actually. So traditionally, as I said, you perfuse with glutaraldehyde the entire organism. The entire organism is chemically fixed from a protein point of view, but for specific reasons, the fixing of the lipids has been only achievable for very small volumes, less than a cubic millimeter, until today, because the work of Sean Mikula, which is one of the groups that is competing for our Brain Preservation Prize, has been working really hard on overcoming that step so that he's a scientist, neuroscientist, neuroanatomist that is trying to map out the circuitry of a whole mouse brain. But a whole mouse brain is huge from this perspective, so he's had to address this osmium tetroxide penetration issue. Mm-hmm. And has shown great results. He's recently published in Nature Methods, and he's got great results showing that you can get a whole mouse brain preserved not as good as the main electron microscopy that people have been doing, but just stunningly good. He may very well be winning our prize very soon. We are in the process of doing evaluation. But this is, irrespective of that, this is a major advancement in the field of electron microscopy, to be able to do an entire mouse brain preserved so that you can do electron microscopy on it at this resolution.
And so would that mean that the next step up would be a human brain eventually?
Absolutely, eventually it would be a human brain. So how I see this going, we need to, there is no use, let me get this very straight out front. Nobody should apply any of these techniques to a human being until they have been checked out with the medical and scientific community, and we have good verification that they preserve the neural structure. Okay, we can argue to a certain extent about what level of criteria should be used for when it is ready to apply to a human being. But I think if you look at the lessons from cryonics, that was applied to human beings way too early, in my opinion. It was applied to human beings way too early. And the scientific community, rightly, had to distance itself from that. And the overcompensation that the scientific community did from those applications to humans was detrimental to the entire human race. Because what it basically said is, now we're not going to do the mainstream scientific research that is required to get this to work, to eventually be applied to human beings. We cannot let that happen on chemical brain preservation. And we need to undo it on chronic preservation. We need to get to a criteria that the scientific and medical community can say, yes, we feel comfortable if people show this level of preservation, this level of reliability of a surgical technique that is being offered to dying patients that want to be preserved for future technology. If they get to this level, then that is a legitimate thing to offer to human patients. And with the Brain Preservation Prize, with the Brain Preservation Foundation that I formed, and the Brain Preservation Prize, it tries to outline what that criteria should be. The criteria shouldn't be that you prove that somebody can come back. Obviously, that misses the entire point. We're trying to get to future technology. We can't wait for future technology to start applying it to people today. But we can insist that we're not creating massive amounts of damage of the very structures that neuroscientists know is crucial to our identity. Mm-hmm. And so we've tried to write the prize in such a way that it has a very rigorous scientific criterion that both groups can agree on. Mm-hmm. And I think we've done that because we've got a bunch of skeptics and scientists on our board of advisors that are looking at this and saying, yes, this is a good prize. We would take cryonics and other things seriously if they're able to meet this, but we don't think they can meet it. Mm-hmm. And we've got the cryonics and teams like Sean Mikula's and others saying, oh, I can meet that prize. Mm-hmm. Yes, we can get there. And so if they can meet at that level, then the next thing that should happen is the medical community should say, oh, we need to actually start thinking about applying this in hospitals, regulating it like any other type of surgical procedure, and we should do it the right way.
So let me ask you this then. I have a few specific details on that whole procedure as it pertains to humans. So first of all, you said that the animal, in this case that you are describing, will be alive, and how painful would that process be for either an animal or a human especially?
Yeah, well, it's not painful at all, because the animal is always put under anesthesia. So I mean, this is a technique that is done thousands of times every day in universities around the world. So and all of the animals have to go under anesthesia before this glutaraldehyde procedure. Mm-hmm.
And how different is the process that you described to the process being used in for shows like Bodyworks, the famous Plasternation show?
Yeah, it's a great question. So it is actually very different. Obviously, there's some superficial similarities, but the chemical, and all you have to ask is when scientists are studying the circuitry of brain tissue, what do they use? They use a specific set of chemicals that are known to preserve those structures that they're interested in. They don't use the bodyworks technique, because the bodyworks techniques, for instance, the most simple thing is that bodyworks does not use glutaraldehyde, so the level of protein fixation is not as strong, and they certainly don't use osmium tetroxide, and so all of the membranes go away in the bodyworks. You don't notice it at a macroscopic level, but at a microscopic level, you certainly would. Mm-hmm. And scientists would never use that. So basically, you want to go back to what neuroscientists use to study the brain tissue. If that could be applied to a whole human brain, that would probably be a pretty good technique.
Mm-hmm. And what do you think would be the relative cost of such a procedure for a big object like the human brain?
Yeah, relative to what?
Relative to, say, cryonics, because I know that, you have neurocryonics projects that are about, say, $80,000 right now or so, if I remember from my conversation with Max Moore, who was another guest on this show some time ago. And then I think if you have a full body cryonic preservation, it was about $150,000 or so.
Yeah. So, what I usually say to that is, we certainly can't give an exact answer, because we don't know that these techniques work. So speculating about it, so there is no protocol that we have today. We can see hints of what a protocol like this applied to a human being would be, but we don't have the protocol yet. It could be that a chemical brain preservation protocol, in order to get really good access to the brain tissue, might require a advanced surgical removal of skull and neuromatter to get into all the crevices, and they have to have this gigantic machine that has thousands of ejection ports to get things in. It could be that it's a $100 million procedure. Now, I don't believe that that's the case. I think that actually it's more likely that it will be much cheaper in the long run. I think it actually could be on the order of $10,000 or something as a surgical procedure. Wow. But we don't know that because we don't have the procedure yet. So it's very difficult to say. The only thing you can say, and cryonics is the same way. So cryonics, the procedure that they use that may or may not work, and certainly doesn't work to a level that we would be satisfied with as perfect, right? They put a price tag on it that goes with what their company can afford. If this was adopted throughout the medical community, there's no reason why cryonics wouldn't also have potentially very low costs. Mm-hmm. So I think the only thing that's really different between cryonics and chemical brain preservation from a price point of view is the storage. And clearly it's going to be easier to store a room temperature object than something that absolutely cannot vary in temperature more than a few degrees, or else problems occur.
Yeah, and the longer the period, the bigger the cost difference, obviously.
Yeah. I mean, one thing I often say, though, is because it would be great to have a low-cost procedure. And I think it would be absolutely great for people to adopt this and to do away with some of the fear of death on a massive scale. Mm-hmm. And I say it would also be fantastic if people started to accept this and incorporate it into their quality of life in a way that says, I I would rather go in for a live chemical brain preservation procedure when I'm 60, let's say, than to wait until I'm 80 and on death's door and have gone through
Alzheimer's.
$1 million worth of taxpayer painful surgeries, and then at the very moment when I'm almost totally destroyed anyway, go in for this procedure. I think that having this as a viable option for people would be fantastic on many different scales. Just one more thing. But talking about the price issue, it is a little bit bizarre because if you have a $10,000 procedure for chemically preserving somebody, we know that bringing somebody back on today's technology would take hundreds of billions of dollars. So it really is somewhat silly talking about it from a price point of view.
Yeah. So you go to a great extent in your paper here, Killed by Bad Philosophy, talking about the procedure too. And, part of the thing that people have to overcome is like their intuitive revulsion, perhaps, of that process, because it looks scary. I mean, people don't like going to the dentist. Now imagine being infused with all kinds of highly toxic chemicals and all that you describe in great detail. So, what do you think would be the difference in terms of public perception and potentially acceptance of your proposal if there were a non-invasive way of preserving the brain rather than the highly invasive method that you're proposing?
Yeah, I'm not
Because
sure
imagine if you have like some kind of a, and I'm not an expert, so I'm totally speculating, but if you can accomplish that kind of resolution without having to slice the brain into pieces, with a very high, fMRI machine or something like that.
That's what you're saying. So you're saying the not the preservation technique per se.
Yeah.
I mean, I think it comes down to,
because the idea is that you're trying to preserve the physical object only so that you get out that information that is stored there. So if we can extract the information, as it is, non-invasively, we don't really care, I think, about the object itself, do we?
Yeah, absolutely. I think that's absolutely correct. I think it would be from a public perception point of view. I agree completely that nobody wants to think about having the most deadly chemicals put into their bodies, even though it wouldn't feel like anything. Like I said, I mean, it's we know how to put people under anesthesia, but the idea of slicing a brain and that is plastic embedded and reading it off and putting it into a computer, it would be much nicer to have a little red pill that you can take and it makes you healthy and live forever. The problem is that these technologies are very difficult. Even this is kind of the zeroth order. Chemical brain preservation is essentially the easiest as it can get from a technological standpoint to preserve what neuroscience says. I think cryonics is harder. Chemical brain preservation is easier simply because it's using a big hammer called chemical fixations, and so to ask, would it be nicer from a public perception not to have to do this? It's kind of like asking, it would be much nicer to have transporters than airplanes, because airplanes crash. You have to, sit next to other people for hours at a time. It's just a pain. Can't we just forget about airplanes? Let's not develop them, and go directly to transporters? No. Because transporter technology is, probably hundreds of years in advance of airplane technology. And it's the same thing I would have to say for any of these other techniques. If we want to get to the future today, we want to have a chance for people today to be put in some type of suspended animation to get to the future of tomorrow, then we've only got two techniques that really come to mind. One is cryonics, one is chemical brain preservation. And I'm happy that both of them are competing for the prize.
So tell me in brief, because we're kind of running out of time here, but in brief, what's your take on the pros and the cons of cryonics as it is today? Because cryonics obviously has a much longer history than your proposal, and in the sense that, well, based on what you said, that's probably a false statement that I just made. Let me reconsider that point. Okay.
Well, they have their own histories. Certainly, it's been applied to human beings longer than the other.
Yeah, and I remember one particular experiment that I watched a documentary on, which very impressed me, and that was about the vitrification of I think it was a rabbit kidney. Mm-hmm. That was vitrified for a couple of days, and then it was surgically transplanted back into the rabbit or a different rabbit, and that kidney was functioning actually, and the rabbit survived for a few days at least. So therefore, sorry.
It was actually a few months. There's a paper on that.
Yeah. So therefore, that was taken as a strong evidence in support of the fact that we are making progress towards better vitrification, and therefore diminishing the damages of ice accumulation and the sort of the freezing process and so on, and therefore we have better chances of accomplishing zero or the minimum amount of hopefully reversible damage that we can incur during the process itself.
Right. So when I try to compare these two, it really comes down to it comes down to electron microscopy evidence. The cryonics community should have. Let's be specific. Alcor, the company that is offering this to human beings as a service. I am a member of Alcor. Okay, I am pay dues. So, as a customer, I would expect to have reams and reams of electron microscopy evidence of what my brain is going to look like if I undergo their procedures, especially if it costs $100,000 and up. They don't have that. They have great reasons for not having that. I think they're great people. I think that they are trying as hard as they can, and the world is against them. Okay? I completely understand that. But at the same time, that is the only thing that, short of a dog getting up and walking off the table after undergoing this procedure, that's the only thing that the scientific community is going to be turned by. If they start showing comprehensive surveys with electron microscopy of cryopreserved brains and saying, here's the level of damage, we will enter a new realm for science of cryonics. Alcor itself has not been has not seen that it is the proper time. It's refused. I don't know how to put it to get into the prize contest itself. But 21st Century Medicine, which is a cryobiology research company that works with some of the same techniques, did the kidney vitrification research that you just described, has been very good in trying to work with us and trying to work with with our requirements for electron microscopy. Mm-hmm. And they've been running tissue samples. They've been trying to get their techniques so that we can look at them in an electron, so they can look at them in an electron microscope and actually show the level of damage. It's an ongoing process. It is the electron micrographs that are out there that I've seen of cryonically preserved tissue do not look normal, but that is not necessarily saying that there was damage. It just means that it looks unlike what the textbooks say chemical preservation looks like, which we know also has artifacts. Mm-hmm. And so we are at this stage in cryonics where there are more questions than there should be, and the people with money that support cryonics, the people with money that would potentially go in for cryonics, they should really think about putting their money into scientific investigation of how good cryonics is. You know, fund a team, fund some independent study of what the quality level is, and tell the rest of the world. And so the more light that we can shine on this, the better from everyone's perspective.
Perhaps they can fund a cryonics prize similar to yours about, you know, the cryopreservation of a mouse brain or something with the least amount of damage, and you put up the factors there that you would accept as, you know, evidence in support of that claim, and let it be.
Absolutely. I mean, people have, that idea has been floating around for a while. And given the fact that we have people that put hundreds of millions of dollars into building private spacecraft, you would think that somebody would say, "Hey, you know, let's figure out what the current state of technology is for cryonics and other alternatives, and let's see if we've advanced from the 1960s, because we certainly have advanced from the 1960s. Anybody who has their mind stuck in cryonics can't work because of this argument that was formulated in the 1960s needs to wake up. It's 50 years later. We've done quite a number of advances over that technology from a biology point of view, and it's time to reevaluate the entire premise, the entire field.
Ken, time is really advancing and working against us seriously here, so I kind of want to go through the last three questions here before we call it a day. You told us that you're a member who pays fee for Alcor, but does that mean that, I take it that means that you do plan to preserve your own brain?
I plan to preserve my own brain if there is a technique that is proven to have a certain level of quality. Mm-hmm. If it turns out in the next few years that Alcor and others cannot live up to the expectations of the Brain Preservation Prize, then I will reluctantly withdraw my support and will reluctantly go back to a normal individual that plans which casket they want. I would like to think that Alcor and or one of these other organizations will be able to meet the requirements of the Brain Preservation Prize within a few years, and so I give them the benefit of the doubt. And until that, I also would like to think that there would be other options that will occur, like chemical brain preservation. If both of them were available and both of them were equally rigorously scientifically tested to preserve the brain structure, I would go for the chemical brain preservation. That's my bias. Mm-hmm. But yes, I think that there is a reasonable possibility that none of this will occur, that nobody will come up with evidence that cryonics works, nobody will come up with evidence that chemical brain preservation works, and that it will never get to a level where it's that rigorous within my lifetime, which, you know, I might die tomorrow for all I know, in which case I will be dead. And that's okay. That's okay. People have died forever, so I'll be just one of those ones that missed it by that much. And that will suck, and I hope that everybody realizes that if they miss it by that much, that it sucks,
and that they should try just a little bit harder, and so maybe my kids will be able to walk on that planet around another star, even though I can't.
Yeah, I don't know about the normal people, but if I'm not convinced that this will work, I would not even want to have a casket at all.
Yeah.
Personally. But anyway, let's go to the last two questions that I traditionally ask of guests on my show. And the second last is this: where can people find more about you and your work?
Yeah, well, the best place is brainpreservation.org. That's the best place.
Mm-hmm. Fantastic. And finally, Ken, do you have a message? The single most important thing that you would like our viewers and listeners to take away from this interview today?
Yeah. I mean, I think the most important thing that that I would want people to take away from it is, we are very close from a scientific perspective to having a technique that can put somebody in suspended animation, so that they can get to future technology. We need to be open-minded in what that technology can be. It is you can't start evaluating it by saying, I'm not going to believe the neuroscience textbooks when they say that we are products of the computations of the human mind. If you start taking science, whole realms of science off the table in your intellectual discussion, that's a problem. And I think a lot of people do that, and they say, well, chemical brain preservation will never work because it requires mind uploading, it requires this other stuff. You need to go back to the science. So I would say if people accept science, accept what the science has to say currently, as being our best guess, may not be perfect, but our best guess about who we are and the way the universe works, then if we tried, within 5 years, we could have a reliable, rigorously scientifically proven preservation procedure for human beings that was offered in hospitals in a regulated environment, where somebody could go in and they wouldn't have this feel of, oh, this is the biggest long shot I'll ever take. It's 0.1% chance that it'll work. They'll have a feel that this is a 99% chance that I'm going to wake up from this. And we could get there in 5 years if we just accept the science and accept what it's telling us about us and our options. And so I think that's, I would ask people to look into it and see where the holes are, but look into it with an open mind and accept what science is saying.
Ken Hayworth, thank you very much for being with us today.
Thank you very much.
Singularity.